Great. Well, it's great to be back here. We were here last year. We got first place last year. Really encourage the judges to nominate and judge based on other startups that need that first place to get funding. We have amazing momentum in our fundraising so far, and wires are hitting like literally every day right now for millions of dollars. So things are going very, very well, and really encourage the judges to look at like what the the scores of these startups are, and then also keep in mind like the need of having an award like this to help those other startups. So I'll get started now. Uh, so this is Maxwell Biosciences. I'm the CEO and founder. Been here just at 8 years, and it was funded by DARPA to create a synthetic immune system long time ago before I bought the patents. The goal here is to restore immunity to reverse disease. So very broad spectrum therapeutics and prophylactics with a novel mechanism of action that targets pathogen membranes, virus, bacterial, and fungi, and avoids the development of resistance. So I'll get into more detail here. We've raised over $27 million in government-sponsored research grants and awards and such, and then $19 million so far in venture stage investment, and we've got a lot more coming in actually this coming week. So forward-looking statements here. Wanted to start with gratitude for Dr. Annalise Barron at Stanford University and Dr. Kent Kirschenbaum at New York University. They are really the father and mother of this technology and pioneered it 10 years before I discovered it and bought the patents. So Kent is our chief scientific officer, and Professor Barron is on our board of directors. We both have significant stakes in the company. So everything you're about to hear today was made possible by the U.S. federal government, South Korean government, specifically U.S. Department of Energy, U.S. Department of Defense, and DARPA, and all of these great institutions that you see here. I also just put this slide up because a lot of what you're about to hear, you're like, wow, this is going to change everything. Can this possibly be true? And we don't do our own testing. We actually send our compounds out to these labs. So they're doing all of this work. This is the data that I'm reporting here in this presentation. So this works in animals and in the lab. Long list of pedigreed investors. Anybody's interested in investing can contact us. We've got a full data room, and you can look at the cap table and those that have invested. Incredible team. We've got the former team from Shire Pharmaceuticals. Their company was just recently sold to Takeda for $61 billion. This guy here was their US head of R&D and is responsible for instance, inventing Adderall, Carbatrol, Equitrol, Claritin, and commercializing those. And so that was responsible for the lion's share of that $61 billion valuation. And then he brought with him the senior vice president, staff's handpicked over into the company. Maxie McFarland is our head of corporate development. He raised 1.4 billion in his last startup as head of corporate development there, a space company. Dr. Tony Verico is here to handle any medical or clinical questions you might have. He's our chief medical officer and has designed our clinical trials. And then here's Dr. Kirschenbaum and director of R&D, Sheetal Valley. So this all started when I saw a study regarding heterochronic mouse parabiosis, where an old and young mouse are connected together so that only the microcapillaries of the skin are able to exchange blood between the two. So it had to be peptides that were squeezing through these microcapillaries that were causing the reversal of aging symptoms in the older mouse. And so the older mouse starts out with weak antimicrobial immune function, high inflammation, weak apoptosis, weakened skin and epithelial tissues, low stem cell recruitment, and low angiogenesis. And of course, when they're connected together, the old mouse gets younger and matches these attributes of the young mouse. And so you can look this up by heterochronic mouse parabiosis. Now, how does that happen? That is the big question. That's the holy grail. Can we reverse human aging by something that we're learning here in these mammals? So this is what brought me out of commercial real estate AI and into biology AI. I funded it, sold my last company to a billionaire, put a lot of money into this, brought a lot of people in to calculate like what is going on in these microcapillaries that is causing this to occur. Also downloaded a bunch of human data from the federal government and ended up with these potential peptide candidates here. What I saw in both sides, both in the aging and youth, was in youth they have more LL37, and in aging humans they have less LL37. LL37 is known as human cathelicidin antimicrobial peptide, one of the most studied peptides in human history, and it's expressed via the CAMP gene. So this is what it's responsible for: antimicrobial, antisepsis, stem cell recruitment, apoptosis, cytokine management, and wound healing and angiogenesis, which is at these concentrations in the bloodstream. So it cleans, soothes, heals, and protects tissues. You see that this is there's a little bit of a logical consistency here where you see that what LL37 is responsible and what we saw in the mice in those published studies is the same, right? So it turns out that LL37 is upregulated, that CAMP gene is upregulated by sunlight, good nutrition, and exercise, particularly via vitamin D. And it does all of these things in these various different organs. I'm not going to go into detail or we don't have enough time. But it's a possible super drug. But the problem is that this particular LL37 breaks down unpredictably and quickly in vivo due to protease degradation. So this is the side chain that's attached to the carbon. And so these carbon bonds right here are what the protease degrade, right? And so if you can get rid of this, then then you can create a stable small molecule, which is what we did. It's no longer a peptide now, it's a small molecule. This is about the size, relative size comparison. Down here we call them clarimers. And now what do we do with it, right? So we decided to go after infectious disease because of the statistical modeling that we were able to download, showing that there's a much larger approval rating with the FDA compared to cardiovascular, psychiatry, and oncology. And when you know the selection biomarker for clinical trials, you have much higher probability of being approved as well. So, infectious disease today is caused by viruses, bacteria, and fungi, sometimes a combination of the three. Often, what clinicians have to say is there's nothing we can do, especially for viruses, fungus, biofilms, and then for antibiotic resistant bacteria, which is unfortunately, a big problem. So, MXB 22510 is the one we're taking into clinical trials now. These are the verified strains of these various different bacteria, fungi, and virus that it's effective against. Remember, these are all done in independent labs. These are escape bacteria, same potency against the escape bacteria as the sensitive bacteria, both gram negative and gram positive. Here's some more compounds that we have and the viruses that they're effective against. I don't have a lot of time because of the technical error here. I have to keep moving. So, pan coronavirus efficacy with another compound that we have. I'm going to skip past. It's essentially pan virus efficacy, not the one that we're taking to the FDA right now. The one that is being researched with the U.S. Army. They're interested for the Ebola efficacy. We had a really great NIH study that showed we had both prophylactic and therapeutic efficacy against COVID-19 disease state. Uh, Totally able to destroy Ebola at 100 micrograms per milliliter, which is a really, really great concentration here. This one is not our antibacterial compound. That's the one that the U.S. Army is interested in. So this is the mechanism of action. It targets phosphatidylserine in the envelope of the membranes of the viruses, which they take from the host cell, the sick host cell, and it puts that marker on there when it becomes infected. So our clarimers target that phosphatidylserine, destroy the virus, and we show no toxicity in animal studies. These are the key compounds. I'm going
Time
to
is
skip.
up, Dr. McClure.
Everything's safe. Let me just skip to the end
Yep.
here.
Yeah, you can wrap up if you like.
Okay, great. And
So that
big
minute you
contract
lost.
with the Army, and that's it. We're raising 20 million on a convertible note. Sorry about that.